Okay, hello students. My name is Arzan. I am a doctor and a lecturer at the Department of Family Medicine. Today, the objectives of today's lecture is what is active immunization and passive immunization, and also I will talk about the type of vaccine. For example, what is whole organism vaccine, what is purified macromolecules, and what is recombinant vector vaccine stands for, and I will talk about DNA vaccine and synthetic peptide vaccine, also multivalent subunit vaccine. We would talk about that. So, what is the passive immunization? Okay, that's the immunization takes from one body to another body. So that means that protection transferred from one person to another, or from animal to the person. So for active immunization is that the natural infections or vaccines can make an active immunization. That means that injected with the virulent antigen can make an immunization process and makes a memory cells, and that's we call active immunization. That's a permanent, that's a stable. So how about the passive and active immunity? To the passive immunity that consists of natural maternal antibody, immunoglobulins, antitoxins. In active immunity, we call that natural infections, and also it included vaccine. For example, the type of vaccine is that attenuated organism, inactivated organism, purified microbial macromolecules, cloned microbial antigen, and multivalent complexes and toxoids. So passive immunization, that's the occur naturally from transfer from maternal antibodies through the placenta to the fetus. Also, there is injection with performed antibodies. For example, human or animal antibodies can be used. Also, injection of animal antibodies can be used also. So the effects of passive immunization are temporary. So how about the challenges of passive immunization? There's three main things. For example, it depends on B cell characteristic signs. For example, B cell defects can be triggered to become immunized. For example, B cell defects can be lead to deficiency of synthesis of antibody. For example, in the second, susceptible person is exposed to disease that will cause immediate complications. For example, there might be some kind of severe complication might become from the vaccine. And number three, the disease is already present. That's the one kind of three main obstacles happen from passive immunization. So how about the common agents used for passive immunization? The diseases that black widow spider white, they use horse antivenin, in botulism horse antitoxin, diphtheria horse antitoxin. How about hepatitis A and B? If there's a human immunoglobulins. How about measles? There's also immunoglobulins. Rabies, also immunoglobulins. Snake bites, horse antivenin. Tetanus, poor human immune response globulin. We use that. So in the immune system, there's some kind of characteristic risks have been influenced on that immunization. For example, antibodies will not trigger the immune system. That's the main thing. There's no presence of memory cells. That sometimes happens, and also there are some kind of risks happen in that passive immunization. For example, recognition of immunoglobulin epitope by self-immunoglobulin paratopes. Also, some individuals produce IgG molecules, and that's why in the person happens mast cell degranulation. Also, in some individuals produce IgG or IgM molecules, and that's why in some person there have been a hypersensitivity reaction. For example, there's an allergy. So the active immunization that will cause from the B and T cell. For example, there's humoral factors. B and C cell plays a significant role in immunization. So formation of memory cell is the basis for relatively permanent effects of vaccination. So principles of vaccination is that there's a self and non-self things. Antigen specificities depends on also indicated by presence of effector cells. Protection from infectious disease using above methods. So there's a schedule what we used in Mongolia. There's a BCG vaccine, hepatitis B vaccine, polio and pentavalent, measles, mumps and rubella, and diphtheria tetanus vaccine. On this schedule, we would talk about future and our practicals. So there's effect how affects the vaccination. It's a effect. It's a so effective. Some vaccines are effective, but some of the individuals from the genetic inheritance, the immunization is not goes on. And also I would talk about the herd immunity. What is the herd immunity? That's the majority of population is immunized, so chance of susceptible individual contacting with the individuals if infection risk of infection would be so low. That's a might be example of measles epidemic. For example, the herd immunity, its factors could be triggered by crowded conditions from the immune system and a higher percentage people with vaccination. It could be a risk factor of herd immunity. So herd immunity is that when people are vaccinated, chance of germ infecting the non-immunized population is small. So what it's that's we have been developed in COVID-19. We have been vaccinated a lot of population in Mongolia, and the herd immunity have been developed, and that's why the infection and outbreak of COVID-19 reduced. That's a benefit from herd immunity. So how about the development of vaccine? How we do that? So the first thing is that it might be comes from the specific branch of immune system, and also it would be developed from the immunologic memory. So what is the role of memory cell? That's the main thing, main key point, key opinion of the immunization. We talk about the memory cell. The memory cell depends on incubation period of pathogen. So the short incubation period, for example, there's influenza symptoms already ended by the time memory cells are activated. Repeated immunization with neutralizing antibodies. What is the long incubation period? Is that, for example, there's a poliovirus, one of the most effective vaccine. It's enough time to allow memory cells to respond. So what happens in the body when they injected with vaccine? There's a serum antibody get peaks and then goes down, but the memory, immunologic memory gets to the height. That's what happens in the body, the levels of the memory. So the type of vaccine, there's a whole organism. It's divided into two parts: attenuated viral and bacterial, inactivated viral and bacterial. So then another type of vaccine is purified macromolecules. For example, polysaccharide, toxoid, recombinant antigen, recombinant vectors, and also DNA vaccine, synthetic peptide vaccine, and multivalent subunit. Whole organism vaccine, what is that? That's a vaccine usually talk about the, for example, in the whole organism that's consists of bacterial cells, for example, anthrax, cholera, pertussis, plague, tuberculosis, typhoid. That's a whole organism vaccine with bacterial inactivated vaccine. How about viral particles? That's a hepatitis A, influenza, measles, mumps, polio, rabies, rotavirus, rubella, varicella, and yellow fevers is referred to in viral particles, whole organism vaccine. Some of them is attenuated, some of them is inactivated. So how about the purified macromolecules? The example is toxoids. How about the disease diphtheria is inactivated exotoxic vaccine? How about tetanus? Inactivated vaccine. And also capsule polysaccharides, for example, Haemophilus influenza type B, and also the Streptococcus and Neisseria meningitis is one of the polysaccharide vaccine we used in the practice. And one of the example of attenuated viral and bacterial vaccine, that's the one example is Basil calmette querine. That's the name of researcher who have been cultivated for a few times, more than 50 times, and then they get less harmless, less virulent vaccine, and that's disease preventing from tuberculosis, one of the most effective vaccine, attenuated bacterial vaccine. So the advantage of the attenuated vaccine, that's the advantage stem from the capacity for transient growth, also prolonged immune system exposure. It is a single immunization and replication within host cell. That's one of the advantage of the attenuated bacterial vaccine. So how about the exceptions? So, for example, in the polio vaccine consists of 3 attenuated strains of poliovirus. Also, colonization of intestine results in immunity to all 3 strains, production of secretory IgA and induction of IgM and IgG. So, result is the need for boosters. Individual strains interfere with one another. So in that opinion, we need to talk about the first immunization, second immunization, and third immunization. What is the first immunization? That's a one-strain predominance and growth. How about second one? Immunity generated by previous immunization limits growth of previously predominant strains. So how about third immunization? That's a simple principle as second immunization. So disadvantage of attenuated bacterial vaccine, that's a major disadvantage is possible reversion. For example, rate of reversion of sabin polio vaccine is in one case in 4 million doses, but it might be a huge dose, but the reversion happens. So the second disadvantage is presence of other viruses is contaminants, and also unforeseen post-vaccine complication. There's a few complications happened after this vaccination. So how about in the future? Genetic engineering techniques provide new methods of attenuation. That's we're looking forward. There's a herpes virus vaccine for pigs is still developing, and also the impossible elimination of reversion is still working on. So inactivated viral and bacterial vaccine. So methods of inactivation include heat or chemical agents, for results loss of replication ability. So difficult to inactivate due to potential of denaturation of epitopes. For example, there's dependence of higher order levels of protein structure. So attenuation inactivation. So what is that? That's a comparison attenuated in inactivated vaccine. Also, we call it attenuated is a live vaccine, inactivated is killed vaccine. So, for example, in the production in attenuated vaccine, selection for a virulent organism. Virulent pathogen is grown under adverse culture conditions or prolonged passage of a virulent human pathogen through different hosts. Booster requirements generally requires only a single dose, but for the inactivated vaccine requires multiple boosters. For example, relative stability less stable, but for inactivated vaccines less stable, more stable. For type of immunity induced, that's produced humoral and cell-mediated immunity. For the inactivated vaccine, that's produced mainly humoral immunity. For reversion process, may reverse to virulent form, cannot reverse to virulent form. So attenuation, that's a normally required dosage to induce relatively permanent immunity, and also primary cell mediated in nature. Despite reliance on cell-mediated immunity, increased IgG response. So what is inactivation? That's the multiple boosters and emphasis on activating humoral immunity. Also inactivation, that's mean cultivated for a long time and get less harmless, less virulent organism. So what is the adjuvant? That's the main topic, main obstacle happens in nowadays becomes because adjuvants, for example, we use aluminium salts is the adjuvant, but this salts is more harmless to the children for the development, especially for the brain. That's the main topic of today's vaccine. So we need to lower those adjuvants. It's because of the harmless effects on the body. So what is the impact of vaccine on public health? It's a really induced the incidence of infections. Also, it's really plays a good role in intervention of infectious disease. For example, between 1977 and 1999, smallpox was eradicated in the United States, but global eradication currently a major consideration. Also, phenomenon of herd immunity is really boosting in public health area. So this is the overall talk about the vaccine, and I would like to provide some links in terms of the vaccine, and please students see that link and get to know about the vaccine in detail. And thank you for your attention.